In the previous episode of Project Gibohagg, we were talking about the first Luca, or last universal common ancestor of all the cells and animals and all the living things on the planet. Now, in this episode, we will be discussing how this last universal common ancestor will diversify into the many forms of prokaryotes and eukaryotes on Gibohagg. So, without further ado, let's go on to the third episode of Project Gibohagg. Luca would be very small compared to the other cells. So one of these was bound to be getting bigger. This rapid increase in size would be so they could eat other smaller cells. They would bend inwards their cell membrane and start enclosing the cell inside of it. After the cell is enclosed, it would start deploying its many vacuoles inside of it, inside the cell membrane onto it. These many vacuoles would just cling to the cell and start producing digestive enzymes to melt the cell, basically. Here's what the cell would look like. We will name this creature multicellular. Many of these cells would be trapped around hydrothermal vents, which are like underwater volcanoes that spew out inorganic materials. Many of these creatures will be chemosynthetic, which means they can produce their own energy through a form of autotrophy, where they take chemicals from the water and separate them. This is what the equation for them would be. Many Earth life can use this to in antioxidant conditions. It's possible that they could also let out hydrogen sulfide. Maybe an offshoot of them could develop the need to eat the hydrogen sulfide being used up by the ancestors. And this is the equation that the hydrogen sulfide eaters would use. These two cells would be identical in the start, but as they keep reproducing and going into different forms, they would start to become very different. Here's what the two cells would look like. The hydrogen sulfide eaters would possibly enclose, surround the methane producers, and as they keep growing and growing, the outside of them will keep growing and growing too. Here's what the colonies could look like possibly. We will name these creatures methanophages and sulfaphages respectively. Maybe one of the cells that eat other cells could start becoming smaller and smaller again. These cells would be so small that they could inhabit inside of other cells' vacuoles. They may develop a flagellum to propel themselves through the water. Inside these vacuole-like structures, there would be something similar to, there would be a substance that they can use to create energy. This is called cell sap, I'm sure. When these cells detect another cell, they would swim up to it. They would then position their sharp flagellum towards the cell. They would then pop the cell open and only have a small window of time after this. And then with this new flexible body, they would position themselves to block the hole up so the cell can't leak out and start sucking up the sap from inside the cell membrane. These could inhabit many parts of the cell too, being found all around it. They would continue to reproduce over and over again until the cell eventually burst open and they would go and look for a new one. Here's what they would look like. We will call these creatures punccellular. One of the methane producers may start getting pushed out of their natural habitat and forced into the upper waters or away from. This would make it so these cells have new opportunities to infect the habitats they inhabit. Here's what the one that would live on the surface would look like. This is because Gibohagg star produces mainly orange, red, and infrared light. These cells may reflect the harmful infrared, red, and orange light and only absorb the yellows, greens, blues, and purples. These creatures would still be capable of asexual reproduction, but they may also develop a sort of reproduction organ to produce more cells. These would absorb atmospheric carbon dioxide and convert it into oxygen. We will name these creatures Rubivo Phocizotor. This is what the one that will be living on the bottom of the ocean would look like. These may live in massive colonies on the bottom of the ocean. They're blue because as you go deeper in the ocean, everything gets tinted to a blue color. So they may try to absorb the oranges, reds, and infrared light and reflect off the blues. They may have two ways. They may have two cells that can absorb the light. One inside of them and one on the outside. That's inside their membrane. We will name these creatures Hycinera Phocizotor. Some hunter cells may start absorbing cells but not killing them. These cells would give protection, lactic acid, and oxygen, and the cells that they eat will give them ATP, or like an energy source. These cells may start maybe not even use like the little enzyme thing that breaks them down anymore. So these creatures will be able to reproduce inside the cells. They may also get smaller because of this. This would also make it so that the ones that give them ATP would have like a smaller genome, and the ones that house them would be able to have a bigger genome. These cells may start absorbing the blue, orange, sulfur, and methane producers and absorbers and stuff. These may also live in giant colonies on the seafloor. Here's what a cell would look like. We will name them Malta and Edentis. Now these creatures start pulling methane, sulfur, and like carbon dioxide from the atmosphere and letting out more oxygen. They may find something similar to snowball Earth on their planet. This would be short-lived though, because an asteroid from the Oort cloud would be able to smash into Gibohagg, ending its icy rule after a short 2 million years. These cells would then start to specialize in many, many, many forms. Come back next time to see what multicellular life may inhabit Gibohagg.